revision video on stem cells and the final modules of the A-level course for AQA. And today we'll be looking at stem cells, what they are, why they're important, how cells go from stem cells to differentiating. So first of all, you need to understand why differentiation is important. So we start off our lives as stem cells in the embryo. Single-celled organisms can never be perfectly adapted because they have to perform all of the functions of their organism, and therefore they can't be adapted for every single thing. However, multicellular organisms can differentiate, and therefore we are able to alter our cells to carry out particular roles. And that's really important from early embryonic stages. So very early on in life, you will start to have heart cells which will beat, you'll start to have brain cells, you'll start to have liver cells, etc. And they all have roles which allow more chance or a more efficient survival as a multicellular organism. In terms of why a cell becomes differentiated, you need to understand that all cells originally in your body came from a zygote, and fertilization occurred, creating this first cell, and then mitosis occurred, creating copies of those cells. At that point, all cells in your body would have been identical stem cells, and in fact now all cells in your body, even today, contain an identical set of genes and DNA. Obviously, we're not including here your gametes, which will have gone through meiosis and have variation. So all your somatic cells, your body cells, will have an identical set of genes and DNA. However, in each cell, different genes are expressed, and that's a word that you're going to have to get quite familiar with. It basically means the genes are turned on if they're expressed, and if they're not expressed, the genes are turned off and don't produce, say, the proteins or the enzymes they are programmed to do. And this lesson that we're going to be looking at now is going to be looking at how different cells are, how different genes are expressed and different ways of doing that. So you can see here at the bottom of the screen why this is important. So you start off with a single cell. Those single cells divide by mitosis to make lots of cells, and those identical cells then will have the same set of 46 chromosomes with all the same genes on them, but this cell at the top is going to be expressing neuron-specific genes, so it becomes say a motor neuron cell. Okay, whereas this one at the bottom expresses epithelial specific genes, so it becomes a ciliated epithelial cell. And therefore that is how differentiation affects, and that affects the organs the organelles they create, and the shape of the cell, etc., etc. So how does this work? Well, some genes are permanently expressed in all cells. And if you think about it, this has to be the case. There are certain enzymes that are needed in all cells. So say, for example, respiratory enzymes, transcription, making of proteins has to happen in all cells. So you need the enzymes that control that, such as RNA polymerase. Okay, membranes are needed in all cells, so you need enzymes that make the membranes. Ribosomes need to be made in all cells. So some genes have to be expressed in all cells in order to maintain that cell being alive. Other genes, however, are permanently switched off in some cells. For example, insulin is a good example. Hopefully you know insulin is produced in the beta cells in the islets of Langerhans of the pancreas, and the beta cells are the only cells that need to therefore have the insulin gene expressed. So the insulin gene is not expressed, for example, in your retina cells or your liver cells where this gene is not being produced. Sorry, this protein is not being produced. And some can be switched on or off as needed, as you'll look at later on in the course. What happens during differentiation? So we go from a stem cell to a specialized or a differentiated cell. Well, cells which differentiate only express some genes. This means that only part of the DNA, therefore, is being translated into proteins during protein synthesis, and only the proteins needed for the function of that cell is produced. If you remember the term proteome, you have the genome, which is the full set of genes in a cell or the full set of genes in an organism, but the proteome is the full set of proteins that a cell can produce, and it's not all of the ones that it has the genetic code for. So it has the DNA code to produce all proteins in theory, but this would be a waste of energy, a waste of investment, and it would mean cells are not working very efficiently. Prevent the genes from being expressed. What does turning off a gene actually mean? Well, there's a few ways that this can happen. The first way is you can prevent transcription. So instead of when the DNA unwinds making messenger RNA, we want to somehow stop this messenger RNA being produced. And the other stage, most likely, is preventing translation. So even if you have made the messenger RNA, is there a way you can reduce the amount of translation that we go into that in a bit more detail, let's think about this. What cells, certain cells are more likely to be able to develop into other types of cells, okay, and some of them are and some are not. So if we look at plant cells, for example, you have two types of cells here. You have xylem and phloem. And this could be an example you're given an exam and asked which one is more likely to develop, to differentiate into another type of plant cell and why. Well, if you think about it, you should know that once the xylem vessel is mature, it has no nucleus, the cell is dead effectively, and therefore it has no genes left over or they're available to use. So differentiation cannot happen in the xylem cells once they've matured because they don't have the DNA available to do so. Differentiation is irreversible in most animal cells, okay, and in fact very few cells retain the ability to differentiate in mature mammals, but the cells that do we call stem cells, and the ones that are in adult cells are adult stem cells, which we'll look at during the rest of this video. So remember, stem cells are undifferentiated cells in animal cells that must constantly replace and therefore are self-renewed. So an example of stem cells in adults would be things like the original cells that become your blood cells, okay, some skin cells, but they're ones that need to be constantly replaced and updated. Look at some sources of some stem cells so you can see here and what uses there might be. So there's lots of different stem cells in cells that are being replaced constantly during your lifetime, and those are important, and diseases, if we could isolate these, could be treated, you can see are very important, everything from Alzheimer's to blindness to heart attacks. Where can we get these stem cells from? And obviously a lot of ethical issues surrounding stem cells. The first type of stem cells that we're going to look at are embryonic stem cells, and 30 cells have been taken for research purposes with consent from 4-day embryos have been shown to differentiate into any type of cell. So we call cells that are from 4-day embryos at the oldest level totipotent cells, meaning they can become any type of cell. If we leave the embryos for 5 days plus, they can differentiate into many types of cells. So after that 4-day cutoff period, some of the genes start to become no longer be able to be expressed, and therefore they go from being totipotent to pluripotent, which is where they can become many types of cells. And effectively, there are two less ethical ways of accessing embryonic stem cells. You can get umbilical cord cells once the umbilical cord is finished with after birth, and there's a lot of cells that are very similar to adult stem cells, and the placenta also can be used to become specific cell types of the body only. Adult stem cells are found in specific tissues in adults, and when we mean adults, really we mean from children upwards, okay, and they are, for example, bone marrow cells and brain cells. They lie dormant until they're needed, okay, and then once they're needed, they will start to proliferate and replace the cells that are being damaged, let's say. They are really difficult to isolate and grow in cultures, but they can only differentiate into some cells or a few types of cells. So there's 3 levels. You've got totipotent, developing into any type of cell, pluripotent, which can develop into many types of cells, and a few types multipotent. And obviously if something's unipotent, it means it is that one type of cell only. So if you took my liver cell, it would be unipotent. Here's just a different slide that I found I thought would be worth showing you. So the totipotent stem cells from a 4-day old embryo can differentiate into all types of cells. Pros are obviously it's easy to isolate and grow, and it can have unlimited cell renewal, but there is a high risk of tissue rejection if you use it from embryos that aren't clones of yourself, and also there are ethical issues. Um, Blastocyst is an example of a pluripotent. So the blastocyst is the ball that the embryo becomes before it starts really specializing, and it can differentiate into cells divided from free germ layers. It's easy to isolate and grow again, but there is again a high risk of tissue rejection, ethical issues, but there is less types of cells that can become, and this is the image here. That is the blastocyst there. You can also get these from the umbilical cord, and something called reprogrammed cells, which we'll talk about in a few minutes' time. Multi-stem cells are for organs with specialized tissues in them. They can differentiate into specialized groups of cells, less likely to be rejected because you're less likely to use your own ones, and therefore there's less ethical issues. But they are hard to isolate, and they are very limited in their differentiation. This then from another point of view. So we start off with totipotent embryonic stem cells that can become any type of cell, and that is really the zygote is the perfect one, which is going to have to differentiate, the zygote being the first cell of 46 chromosomes in your body that is being made from the egg and the sperm fertilizing. That is going to then be able to differentiate into all cells because that's where you have come from. We then go on to pluripotent embryonic stem cells, where embryos can differentiate into almost all types of cells, embryonic, and you get these from embryonic cells and fetal stem cells. And you can see here they become one of 3 types of cells as the body develops as a result. And then adults stem cells can differentiate into limited numbers, so we call them multipotent, normally because the cell is a particular type. So for example, bone marrow will always become blood types. We can, however, treat adult stem cells and make induced pluripotent stem cells, which we'll talk about later on in the video. And once you get to certain cells, unipotent cells will only differentiate into a single type. So that's when they become specialized as a result. So if you take heart cells from the heart, it will become heart cells only. You'll be aware from GCSE is the ethical issues to do with them, and we want to be able to get stem cells from ethical sources, but be able to use them for these amazing potential disease cures that we could potentially have. And one way of doing this is by doing something called induced pluripotent stem cells. And basically what this is about reprogramming adult cells to become any other type of cell. So we can take an adult unipotent stem cell and turn it into induced pluripotent stem cells. Now, this is obviously a really complicated procedure, but in terms of what you need to know for A level, it is very limited and basic. So all you need to know is that you would take a unipotent cell, which is almost any cell in your body, and you would genetically alter it to give it the characteristics of embryonic stem cells. You don't need to know the details of that. Effectively, what you are doing then is turning on the genes, because remember, every single cell has a full set of genes. So we're turning on the genes that are not expressed, and we do this using something called transcription factors. And transcriptional factors are things that which will promote transcription of that part of the gene. They're often proteins, but we'll talk about them in the next video. These induced pluripotent stem cells have some new characteristics, so they will be able to do the things you'd expect, but also they can have self-renewal. So what this means is that once you've worked out how to do it, you can in theory keep making limitless supplies of this. So for example, if you had a damaged pancreas, in theory you could create more pancreatic cells and continue being made on a conveyor belt, for example. If this was successful, we could complete medical research without the use of embryonic stem cells, and that would avoid the ethical issues which would be significant for medical enhancements. So just to sort of summarise it then, we have and how stem cells are being used. Embryonic stem cells have been used in areas, but their use and research is 100% banned currently in the US. In Europe, again, their embryonic research is 100% banned since 2008. In the UK, research is approved on individual basis. Induced pluripotent stem cells are when somatic body cells have already differentiated. You can add different genes from a mouse embryo, which controls the pluripotency, and this codes for transcription factors which turn the gene back on. Side effects, though, where this has been experimented with, it has led to potential inducement of cancer, so that's not good. Potential benefits, though, could we use to grow and re-damage tissues from accidents? We could use it for skin grafts, for burns, and we could potentially damage tissues from diseases such as Parkinson's disease or multiple sclerosis, which is when your own nerve cells are damaged by your own white blood cells. So it's an autoimmune disease.